So hello and welcome to this webinar. We will be talking about antibody screening and characterization using LabKey's Biologics LIMS. Thanks again so much for joining. My name is Bernie Lee. I'm a product manager here at LabKey Software for our Biologics LIMS. So I want to spend most of our time together today actually demonstrating some aspects of the product. But first, I want to set the stage just a little bit, since there's very likely to be some people here who aren't actively doing this kind of science. So I want to make sure that we all in general understand what's going on. We're going to be using some screening and characterization operations that are common amongst discovery campaigns in the early stages to demonstrate. But I'll briefly set the stage here. So screening and characterization are some early steps in that discovery and development for antibodies, as well as other large molecules that will be used for therapeutic, diagnostic, and other sort of research uses. And they ensure that only the most effective antibodies progress to later and often more expensive stages. That screening side, the most common, I'll say, purpose for doing that screening is to go from a huge number of possible sequences of potential antibodies that down to many fewer that specifically bind to some target antigen, and also that don't bind too heavily to similar antigens. And the process involved generally is one in which you screen and enrich sequence libraries, and then you often use quantitation methods like ELISAs and flow cytometry and other sorts of potential high throughput methods to quantify those interactions, and then to select candidates with high specificity, high affinity to the target. And the outcome is the identification of candidates that you want to further analyze, that you want to further develop. And a lot of that analysis is characterization. It's the generation of a profile for the selected set of candidates to further assess their functionality, their biological activity, and what boils down to their suitability as candidates for further development. A couple of examples of parameters that are assessed: binding affinity, we already talked about. If you have an antibody that barely binds to an antigen, go ahead and toss it out. Functional assays, stability and specificity. Again, all of these things ultimately really matter. They are great ways of starting your campaigns to filter down from millions, billions, or more possibilities to a human manageable quantity. Okay. So there are some challenges when you do this, and probably the biggest one is the high volume. These large scale screenings, they generate huge amounts of data. There's a lot of samples and plates. There's a lot of assay runs. And there's a lot of data to be integrated and used. And managing and analyzing large amounts of that data manually in spreadsheets can be risky. Mistakes are definitely made, and it's certainly time consuming. And leads to organizational issues with different people having different files containing repeated information or missed information. Not great. Integration, integrating data from all the different samples you're dealing with in different locations and different plates, across platforms like ELISA, flow, and surface plasmon resonance, that can be challenging. And you can end up with different data formats, inconsistent annotations, and when you find yourself in a situation like that, analysis and proper interpretation can be harder. There some quality challenges. Things like inconsistent and incomplete data can lead to false positives or negatives in your analysis and selection. And while, when you're going from, millions to billions of candidates down to a few, you're using broad strokes, you still don't want to miss things that are potentially good unnecessarily. Then ensuring data accuracy and reproducibility across the different assays and experimental setups is, of course, critical. These are multi-step processes. You don't just do one round of screening and enrichment and then quantitation. That would be, the luckiest discovery campaign of all time. These are complex multi-step operations. They're very iterative. There are multiple rounds where you do things in a more stringent way, or you quantitate differently so that you get different data to paint the picture. Managing these steps efficiently while tracking samples through their different plates and locations can be quite difficult. And then assay variability. There are, of course, variations in assay conditions that can impact the consistency of results. And so it can be really valuable to standardize protocols, standardize data shapes across labs, across groups, across projects to help ensure accurate analysis. Okay, I hope that has set a reasonable stage for what I'll be showing you today. Obviously, if at some point there are further questions or details, as I hope most of you have seen in the chat, we invite you to make this interactive. Please put some questions in the chat or in the Q&A for the webinar, and we'll try to talk about those things. So, LabKey's Biologics LIMS is a very powerful product that is as complex as the science that it aims to serve. And I'm going to show you a few aspects of the product today in order to paint a picture regarding the way that you can do some of this kind of screening and characterization stuff. So I'm gonna start by showing you a little bit about our bioregistry, which you can use to profile and track known entities, which are ultimately generated throughout the process. We're going to talk a bit about sample and plate management, including tracking through the various plate operations, like the screen and enrichment rounds. And we'll talk a little bit about capturing assay data, performing analysis and hit selection, and in general what that looks like. So with that, we're gonna switch screen share here, and we are going to look at LabKey's Biologics LIMS. Okay, so I'm starting us on, oh, 1 second. There we go. I'm starting us on this molecule overview, okay? There are a lot of aspects of this product. There are a variety of other entities in our bioregistry and more that you can add. But you can track individual sequences, libraries, cell lines, the vectors that you end up using. You can have a variety of sample types that you are tracking, lots of different assays that you might use to characterize these materials, plates, storage, media, notebooks, lots of aspects here. I'm not going to get to most of them today. Like I said, what we're going to start with is this molecule overview. So registering entities of interest like a molecule and its components is a really valuable way of having a single source for tracking and reporting information as it's generated. So when you are starting a campaign, you don't already have a commercial name for an antibody. I'm using this purely to paint a picture. But in our molecule registry, you can capture all kinds of information about what you know about it and add more later as you go. In this case, our system automatically classifies this molecule as an IgG Fab based on the structure that was used to register. And in this case, that's a 1-to-1 Fab heavy chain light chain combination, which are also entities tracked in the system. You can also see a variety of physical properties calculated for this molecule, which might be used for analytical purposes. And all of these things are interrelated. So, this Herceptin Fab light chain sequence is a real entity in the system. I can click through to it and I can see, the particular chain formats for this molecule. I can see its particular physical property calculations. I can actually come and look at the sequence itself and see the way that it's annotated with various frameworks and CDRs. And I can see the way, again, that it's connected back to this molecule in the system. This molecule also has information about all of the associated samples that you might have generated. In this case, there are some transient cell lines that contain the construct, the plasmids that are used to produce this molecule. There are some protein batches expressed by those expression system and some purified antibodies. So this implies that there is some downstream work that has been done on some of this stuff beyond the initial screening and characterization. And I have one notebook here that is clearly making reference to work done on this particular molecule and is referenced here. Okay. So getting this kind of information out of spreadsheets, centralizing it in some kind of data management system like LabKey's Biologics LIMS can help you maintain the uniqueness of some of these entities that you're scrutinizing, as well as ultimately increase the efficiency of exploring the data that you have, planning work, and reporting on it. So I showed you how these sequence chains are associated with this molecule. We actually generate significant graphs that show all of these relationships. So here's my Herceptin Fab, and you can see those heavy chains and light chains above. You can also see the expression systems that are producing these things, and all of the samples that are coming out of the other end. So these are some transient cell lines. These are batches of this molecule being produced, and you can see at the bottom here we have some purified antibody that's generated for these things. So as you work through the system, registering relevant entities, we will not only help you keep track of the samples and the data, but we will construct these relationships for asking questions of the data. Okay. In addition, any characterization, any assays run on the materials associated with these entities, we will roll up into a profile for you. So, I showed you the various samples that have been generated in association with this molecule. On the assay results for this, you can see that for one of these batches, we did some chromatography. Here's a very generic anion exchange profile. We did some protein quantification. There's a page gel associated with some of this stuff. Any assay that you run on samples associated with this molecule can be rolled up so that at any point in time, you can come back and examine the full profile that you've generated for these entities. Okay. So how did we get here, and how do we do some of that initial screening and characterization? So I created, I'm tracking some libraries in here, and I have generated some stocks of these materials. Okay, and this is obviously very contrived, but I have a set of glycerol stocks that's what GS is, glycerol stock, of these materials that are sitting in my system. And we have a lot of very rich sample management faculties in our biologics LIMS. You can see that these samples have volumes, free thaw counts, expiration dates, if that's something you want to track. You can see where these are in storage, the locations who put them there. You can see information about aliquots generated. All of that information is available to you for tracking, and this is something that we can dive into more later. But when there are a set of these stocks that I want to begin screening for, I can simply take them and I can say, I want to get started on this, and I want to add these to a plate set. So I can choose where to stick these things, and I can hit go. I'm not going to walk through each of those steps right now. I have this pre-fabricated for us. So here is those glycerol stocks, 1 through 94, set it on a 96 well plate. We support this at much larger scale than this. If I wanted 20, 30, 40, 50 of these, that's fine. And we would simply populate this plate set with a list of all of those of interest. I can capture a variety of pieces of information about these samples in their wells if I care to. In this case, what I've simply done is shown the reference to the library that I'm keeping track of, and the volume that I'm putting in these plates. Okay. And so from here, from this primary plate set, from this stock of glycerol stock, I can then begin the screening and enrichment process. And so I can take all of these samples, and I can put some quantity of them into new locations for quantitation, or simply to pass them between the screen and enrichment phases. I can stamp some quantity of these onto a new plate in order to do work on them. And as I do so, what we do is we will construct this plate set hierarchy in order to maintain the associations between these plates and the materials on them. And then as I generate data for these plates, we roll all of that information up. So in this case, I might do some of that initial screen and enrichment, maybe 2 or 3 rounds of it. And then at some point, what I would do is I would pull some of that material out to quantify the binding of what's left. So in this case, I grabbed a small volume of the remaining material in those wells to run an ELISA against a particular antigen. Now, this antigen is actually a registered molecule in the system. I could show you that in a minute, but in the same way that we were looking at that Herceptin fab before, if there are molecules that you use as things like antigens, you can have those registered as well, and you can capture information against them where you've used them in assays and things. So once I have laid these samples out of my plate and captured whatever information is relevant to me, I can simply import my plate reads. Now I have whatever this is, 7 or 8 plate-based assays configured in here. And importing this data is as simple as choosing that ELISA and then dropping my plate read right here and hitting go. And what we will do is automatically make the association between the individual well result and our plates and the samples that are there. Oops, clicked on the wrong spot. But once I import those, then the results become available for this plate. Okay, so here are those imported results. I have positive binding and I have off-target binding. And from these raw results, there are a variety of things that I can do. I generated a bar chart representation of the positive binding of these wells. I also generated a plate heat map that shows positive binding per location per sample. But of course, what you want to do from here is do your analysis, do your selection, and very often in these early stages, that's as simple as having thresholds. So As I change these thresholds, for example, if I want positive binding always to be above 0.3, then I can set that threshold, and the visualization updates accordingly. I'm actually gonna back out of that because I have these prefabricated. I have a 0.1 threshold and a 0.3 threshold. And when I choose those, what automatically happens is I get the samples for this ELISA run that meet my criteria. Oops, although I appear to have messed that up. Okay, so what that leaves me with is these 51 samples that meet my criteria. And what I can do is then take these, although take these and mark these as hits. Okay, and this is valuable because even though I ran this assay on material extracted from my original stock, if I go back to that original primary plate, I actually have that same data rolled up onto here. Okay, so these are the hits that I marked according to the threshold analysis that I did. And that allows me to review each individual assay that I have data for, and then make choices about what to pick going forward. Okay, so I can look across each of the individual analyses that I've done, and I can choose the ones that meet all of those criteria. I can choose them, and I can pick them forward. I can also review those particular assays if I'm interested in, but this is a way of integrating data from down that whole plate set hierarchy, which can be many steps long, and making it really easy to say, okay, the material that was wherever this sample was on this plate where I ran titer, or this plate where I ran ELISA, or some plate where I put it through some flow cytometry analysis. Of course, for an alpha ELISA, you have that same sample in lots of different places. What we will do is roll it up for you so that it's really easy for you to know exactly where that material is on your primary plate, so that you can then take that material and use it in further work. One other thing I want to point out really quick, for this alpha ELISA, I have automatically generated EC50 values and calculated max values for the raw data that's present. I actually only put 3 of those samples through. And right now I'm showing you a filtered view of this, but all of the raw luminescences for each of the places that these samples were present at different concentrations is there. And the calculations are automatically generated for you when you import the data. And we can make it present for your analysis here. Okay. I hope that paints a picture generally of how you can walk through initial screening and characterization of some of these materials. All kinds of other sample operations, discovery and development operations, are supported in this system. But I just wanted to give that initial walkthrough. I'm going to switch back over here. Okay, just to reiterate a couple things really quick. So I showed you only a few aspects of our biologics LIMS. I wanted to make this succinct, but I also want to re-emphasize some of the main benefits that come from the efficiency gains that in streamlining your antibody discovery and development. So the first one that I really like to harp on is this reduced dependency on spreadsheets and a centralized place, in a centralized place, which reduces error and the time required to track and report on your operations. You can track and maintain uniqueness for entities. So I showed you a molecule that has a particular identity defined by its components in this case. Those 2 sequences, stoichiometries of 1 each. We will make sure that you don't register something else with the same configuration. Those sequences themselves have a sequence identity according to their amino acids or based on their amino acids. And we will help maintain the uniqueness of those things so that as you as you go through the discovery process, if you find that you actually have two of the same sequence or two very similar sequences, it's very easy for you to choose just one of them and continue forward. And of course, the integration of the information that you're capturing through those iterative plate operations really helps facilitate decision making. Much of the data capture and analysis can be automated. LabKey can listen for new data that you're generating, or you can simply throw your plate reads and other results at it, and will automatically associate the right data with the right well, right sample, and ultimately the right entity upstream of that. And we will also generate those calculations and visualizations that you need for your analysis and selection automatically. There are many other aspects of Biologics LIMS that I didn't show you today. Things I didn't dive into freezer management. I didn't dive into media tracking. But I do want to briefly call out our data-connected electronic laboratory notebook that's built in so that in the same system in which you're doing your work, capturing your data, doing a lot of your reporting, you can comprehensively author and report your findings as well. So we at LabKey, thank you very much for joining us for this initial tour of LabKey's Biologics LIMS. I'd really like to take a deeper dive with you. There's a lot more to learn. This is a very adaptable system for the variations that we see continually in research. And so I'd love to follow up with each of you to talk about your use cases, your pain points, your goals. So keep an eye out for an email from me that has a link to schedule some time. And what we can do there is talk about those aspects, and I can prepare a more tailored demo for your organization. I do see that we already have some questions, and I'm very grateful for that. If anyone has any more that they'd like to ask here, as opposed to saving some for a future discussion, I'd be delighted to talk about them. So again, thank you, and let me have a quick look at some of these questions. Okay, I see a couple here. What happens if there are replicate values from a particular assay? Are the data averageable? Yes. So as you saw in that alpha ELISA, we will retain the raw values for you. But if you have replicates, you can both maintain the original raw values, and we can also automatically on import generate like a mean and standard deviation for those values from which you can do the rest of your analysis. So you can have those means be what shows up in your visualizations and your calculations. You can also examine the raw ones if it's relevant to you. And then I see knock out bad values from an assay. So QC exclusions, things like that. Yes. So if you import some data and then you have a look at it and you say, assuming the run as a whole is in good shape, which you can set up QC states so that when data first comes in, you have the ability for you or one of your scientists to say, hang on, there was something wrong with this run. I don't want it submitted for other people to be able to see it and make decisions based upon it. But once it's past that state, you'll still be able to say, this one replicate is like a massive outlier. We have no idea what happened. Or maybe you can go back in the plate and look and see that you had massive evaporation and your measure was way out of bounds. You have the ability to exclude that, to mark that one as excluded, and then those replicate calculations will be rerun. And so you'll still see that there was a result, but it won't mess up your analysis. Thank you for those questions. Let's see. Are there any others today? Like I said, I'm happy to answer as many as there are today. But if you're interested in a deeper dive with further questions on an individual basis, we would love to set up that time. If there are no other immediate questions, again, thank you so much for your time and attention today. We'll look forward to hearing from you.